A misty morning in Bavaria, 1954. The peat diggers pause, shovels suspended mid-air. Something gleams in the dark soil, not gold, but bone. Human bone. Ancient human bone. When scientists arrive, they discover not just any skeleton, but one with a genetic signature that would perplex researchers for decades. This signature contains markers never before seen in European remains, markers that seem to emerge from nowhere, with no clear migration pattern to explain them. How could an entire genetic lineage appear without leaving traces of its journey? Asked Dr. Renata Dietrich, carefully brushing soil from the cranium. It is as if they materialized from thin air. This was just the beginning. Germany, this heartland of Europe, would continue to yield genetic mysteries that defy conventional explanations. While most European populations tell straightforward stories of migration and mixing, German DNA holds puzzles that have geneticists rethinking the entire narrative of European settlement. From Neanderthal enclaves to mysterious maternal lineages that match no known migration route. From lost tribes to genetic markers appearing centuries before they should, Germany's genetic landscape is unlike any other in Europe. What if everything we thought we knew about European ancestry was incomplete? What if, hidden in the genes of modern and ancient Germans, lies a story that rewrites human history? Today, I will take you on a journey through time, from the Ice Age to modern genomic laboratories, as we unravel what might be the most perplexing genetic mystery in Europe. What makes German DNA so uniquely enigmatic? Why does this seemingly central European population harbor genetic secrets found nowhere else on the continent? And what might these secrets reveal about all of us? The story begins not with humans, but with mammoths. In 2017, in a cave near Heidelberg, paleogeneticist Dr. Olivia Brenner made a discovery that would upend conventional wisdom about ancient species interactions. Analyzing sediment DNA, genetic material left behind in soil, she found something impossible. Mammoth DNA dating to just 9,500 years ago, a full 5,000 years after mammoths were thought to have disappeared from mainland Europe. We ran the tests 3 times, Dr. Brenner explained. The dates kept coming back the same. Somehow, in this isolated region of what is now Germany, mammoths survived long after they vanished everywhere else in Europe. This anomaly forced scientists to question. What was different about this region? Why would megafauna persist here when climate change and human hunting had eliminated them elsewhere? The answer came not from the mammoths themselves, but from human remains found in the same area. The Heidelberg humans carried genetic markers indicating they abstained from hunting mammoths, despite hunting other large game. Further analysis revealed ritual objects made from mammoth materials, not taken through violence, but collected after natural deaths. This suggested a cultural taboo against mammoth hunting, possibly a form of proto-conservation unknown elsewhere in Ice Age Europe. More remarkable still was that these humans carried genetic markers previously only found in Siberian populations, suggesting a migration that brought both cultural practices and genes that had bypassed the rest of Europe. But how did this isolated population arrive in central Germany without leaving traces in intervening territories? It is as if they did not walk, but flew, remarked Dr. Rudolf Dahl, comparing ancient DNA samples in his Berlin laboratory. We see no genetic breadcrumbs between Siberia and Heidelberg, just 2 identical signatures separated by thousands of kilometers. This mystery deepened when researchers expanded their search to other ancient German sites. Throughout central Germany, they found similar genetic anomalies, populations appearing with established genetic signatures but no clear migration route. It was as if Germany had served as a quantum genetic jumping point, where populations materialized with fully formed cultural and genetic identities. To understand why this matters, we need to step back and examine how human migration typically works. When populations move, they leave genetic traces like breadcrumbs in a forest. They interbreed with local populations, creating gradient patterns that geneticists can trace backward, reconstructing ancient movements. This is why we can follow the spread of agriculture from the Near East or track Indo-European languages through DNA. But Germany breaks this pattern repeatedly throughout prehistory. It seems to have served as a genetic wormhole, where populations appear fully formed, challenging our understanding of how humans spread across continents. The next piece of our puzzle emerged in 2019 from an unlikely source: ancient teeth found at multiple archaeological sites across Germany. Dr. Brenner, now leading a pan-European genetic study, discovered something astonishing. Genetic adaptations for digesting starch appeared in German remains centuries before the arrival of agriculture. These adaptations should not exist without agricultural practices driving natural selection, she explained, pointing to genetic charts in her laboratory. Yet here they were, in hunter-gatherers who had never encountered domesticated grain. It was as if their bodies were preparing for a future they could not possibly have known was coming. This genetic foreshadowing defied evolutionary logic. Natural selection works backward, not forward. Organisms adapt to existing conditions, not future ones. Yet somehow, ancient German populations developed genetic adaptations for an agricultural lifestyle before agriculture arrived. Some scientists suggested these populations might have practiced proto-agriculture, cultivating wild grains at a small scale. But archaeological evidence contradicted this theory. No cultivation tools or domesticated plant remains were found in these early contexts. "We are left with a genuine scientific anomaly," admitted Dr. Dahl. "Either our understanding of natural selection is incomplete, or something extraordinarily unusual was happening in prehistoric Germany." This genetic time paradox becomes even more perplexing when we look at linguistic evidence. Modern German contains phonetic elements and grammatical structures that linguists have traced to languages spoken thousands of kilometers away in Central Asia. These elements appear in German with no clear diffusion pattern through intervening territories. Dr. Elena Rossi of the Max Planck Institute for Evolutionary Anthropology suggested a radical hypothesis. What if Germany represents not just a geographical crossroads, but a temporal one? A place where multiple timelines of human migration somehow converged. While most scientists reject such speculative explanations, the data continues to defy conventional interpretations. The German genetic landscape resembles a puzzle with pieces from different boxes, assembled into a picture that makes no sequential sense. Let us now fast forward to the Bronze Age around 4,500 years ago. This period saw massive population changes across Europe, with migrations from the Pontic-Caspian steppe bringing new languages, technologies, and genes. These migrations followed well-documented routes, leaving clear genetic signatures from east to west. Yet once again, Germany presents an anomaly. In 2020, archaeologists excavating Bronze Age graves near Munich discovered individuals with steppe ancestry, but without the typical genetic markers found in other European populations with steppe heritage. It was as if they had taken a different evolutionary path, explained geneticist Dr. Marco Schutz. Same starting point in the steppes, but a completely different genetic outcome than their counterparts who settled elsewhere in Europe. This discovery suggested that something in the German environment, either physical or cultural, was exerting unique selective pressures on incoming populations. Whatever this factor was, it seemed to accelerate genetic divergence, creating distinctive lineages that appeared nowhere else. The most compelling evidence came from mitochondrial DNA, genetic material passed exclusively from mothers to children. Several mitochondrial lineages found in ancient German remains have no clear origin population. They simply appear, fully formed, without the expected ancestral transitions. It is like finding a novel written in a mature, complex language, but with no earlier texts showing how that language developed, Dr. Brenner explained. These mitochondrial lineages had to come from somewhere, but that somewhere has vanished from the genetic record. Some researchers proposed that these lineages originated in populations that were later completely replaced, leaving no modern descendants except through these German lineages. But this explanation fails to account for why this pattern repeats throughout German prehistory and nowhere else with such frequency. The mystery intensified when researchers compared ancient DNA from Germanic tribes with that of neighboring Celtic and Slavic peoples. All three groups had originated from similar founding populations, yet Germanic tribes showed accelerated genetic differentiation, as if their evolutionary clock ran faster. This accelerated evolution wasn't random. It clustered around genes involved in immunity, metabolism, and neural development. Whatever was driving this rapid change appeared to be preparing Germanic peoples for specific environmental or cultural challenges. Dr. Dahl proposed what he called the crucible hypothesis, the idea that Germany's position at the crossroads of European migration created extreme selective pressures. Imagine a population repeatedly exposed to new pathogens, new cultural practices, new dietary options, he suggested. Such conditions might drive unusually rapid adaptation. Yet this hypothesis fails to explain why other crossroad regions, the Balkans, for instance, don't show similar patterns of accelerated genetic change. Something about the German geographical or cultural context was uniquely transformative. The Roman historian Tacitus, writing in 98 CE, offered an intriguing clue. In his ethnography Germania, he claimed the Germanic tribes were indigenous and unmixed with other races. While modern genetics disproves this claim of genetic isolation, Tacitus might have been observing the results of this accelerated differentiation. Germanic peoples who had diverged so rapidly from their ancestors that they appeared distinctive even to ancient observers. Tacitus might have been witnessing the outcome of this genetic mystery without understanding its cause, suggested historical geneticist Dr. Sandra Meyer. These weren't pure peoples, but rather populations transformed by unknown selective forces. As we move forward in time to the migration period, roughly 400 to 800 CE, the German genetic landscape becomes even more complex. This era saw massive population movements across Europe, with Germanic tribes expanding outward while other groups moved into traditionally Germanic territories. Genetic studies of remains from this period reveal something unexpected. Despite enormous population upheaval, certain genetic signatures remained remarkably stable in German regions. It was as if something about the geography itself was selecting for particular genetic traits, regardless of which population inhabited the area. We see ecological genetic convergence, explained environmental geneticist Dr. Thomas Weber. Different populations moving into the same German regions begin to show similar adaptive markers within just a few generations, much faster than natural selection typically works. This rapid convergence suggests that something about the German environment, perhaps its water sources, soil composition, or native microbiome, exerts unusually strong selective pressure on human genetics. Whatever this environmental factor is, it appears to have remained constant for thousands of years while transforming the genetics of each new population that encountered it. The most tantalizing evidence comes from a 2021 study comparing ancient pathogen DNA from archaeological sites across Europe. German sites consistently showed unusual pathogen communities, including several microorganisms that appear nowhere else in the ancient European record. These unique pathogens might have driven unique human adaptations, proposed Dr. Brenner. If German populations faced microbial challenges not present elsewhere in Europe, their genetics would have evolved along distinctive paths. This pathogen hypothesis gained support when researchers discovered that many of the accelerated genetic changes in ancient German populations involved immune system genes, particularly those responding to viral infections. Whatever was shaping German genetics seemed to be, at least partially, microbial in nature. Yet pathogens alone cannot explain all the genetic peculiarities. Some of the most distinctive German genetic signatures involve metabolism, brain development, and even behavioral predispositions, traits unlikely to be shaped solely by disease resistance. We're dealing with multiple overlapping selective forces, suggested Dr. Dahl. Pathogens, diet, social structures, and possibly factors we haven't even identified yet, all working together to create this genetic outlier effect. As we approach the medieval period, another layer of the mystery unfolds. Historical records document extensive German eastward expansion during the Ostsiedlung, the medieval German migration into Slavic lands. Genetic studies of modern populations in these regions reveal something curious. While German cultural and linguistic influence was profound, the genetic impact was often minimal. It's as if the Germanic genetic signatures were fragile outside their homeland, noted population geneticist Dr. Annika Fischer. Once removed from whatever environmental factors shaped them, they were quickly diluted through intermarriage with local populations. This pattern suggests that whatever made German genetics distinctive was tightly bound to the German geographical context. When Germanic peoples migrated outward, these unique signatures faded within a few generations, overwhelmed by the genetics of their new homes. The exception to this rule appears in isolated German-speaking communities, that maintained not just cultural but environmental continuity with their homeland, such as certain Alpine settlements and Baltic German enclaves. These populations retained distinctive German genetic signatures much longer than those who fully integrated into new environments. The genetics seems to follow the environment more than the culture, observed Dr. Weber. German speakers who recreated German agricultural practices, architectural styles, and diets retained these genetic signatures longer than those who adopted local lifestyles. This environmental connection brings us to perhaps the most controversial aspect of German genetic mystery, the possibility that certain regions within Germany represent what geneticists call microevolutionary hotspots, locations where human evolution proceeds at accelerated rates due to unusual selective pressures. Several studies have identified regions in Bavaria, the Rhineland, and parts of eastern Germany where genetic diversity doesn't follow expected patterns. These regions show unusually high rates of novel genetic variants not found elsewhere, as if these areas were genetic innovation incubators. These hotspots challenge our understanding of how human evolution works, admitted Dr. Brenner. The conventional timeline for new genetic variants to appear and spread through a population is tens of thousands of years. In these German regions, we're seeing it happen in centuries. This accelerated evolution isn't producing dramatic physical differences. Modern Germans are not evolving into a separate species. Rather, the changes involve subtle metabolic, immune, and neurological adaptations that might not be visible to the naked eye but significantly impact how bodies function. Some scientists have proposed that these microevolutionary hotspots might be linked to geological factors. Germany has numerous mineral springs, distinctive soil compositions, and unique geological formations that might influence local biology in ways we don't fully understand. We know that trace elements in water and soil can affect human health and development, noted geobiologist Dr. Martin Schmidt. Perhaps certain German regions provide unique mineral combinations that have influenced human evolution over thousands of years. This geological hypothesis gained credibility in 2022 when researchers discovered correlations between genetic markers in local populations and the mineral content of regional water sources. Areas with distinctive mineral profiles showed correspondingly distinctive genetic adaptations related to mineral processing and detoxification. Yet geology alone cannot explain the full scope of German genetic distinctiveness. Cultural practices, particularly those involving food preparation, childcare, and social organization, also shape genetic selection. And here again, German regions show unusual patterns. Anthropological records document distinctive fermentation practices, food preservation methods, and dietary combinations in traditional German cuisines. Many of these practices alter the nutritional and microbial content of foods in ways that might exert unique selective pressures on the populations consuming them. Diet shapes genetics, which shapes culture, which further shapes diet, explained cultural geneticist Dr. Lena Hoffman. In Germany, we may be seeing the results of thousands of years of this coevolutionary cycle operating in isolation from broader European patterns. This cultural genetic feedback loop might help explain why certain German genetic signatures persist despite massive historical population movements and admixture. The cultural practices traveled with the populations, maintaining the selective pressures that had shaped their genetics in the first place. The medieval period adds another layer to our mystery. During this time, Germany was not a unified nation but a patchwork of small states, ecclesiastical territories, and free cities. This political fragmentation created numerous small, semi-isolated breeding populations, a perfect laboratory for genetic differentiation. Medieval German principalities functioned almost like evolutionary islands, noted historical geneticist Dr. Klaus Wagner. Small population sizes, limited exchange with neighboring territories, and distinctive local environments accelerated genetic drift and local adaptation. Studies of medieval cemetery remains confirm this pattern. Genetic markers often cluster tightly by historical territory, with clear differentiation between neighboring principalities despite their geographical proximity. This genetic fragmentation persisted well into the early modern period, creating a genetic landscape unlike anywhere else in Europe. Most European regions show gradual genetic gradients. Populations blend into one another across geographical space, with no sharp genetic boundaries. Medieval Germany, by contrast, was a genetic mosaic, with distinctive population clusters separated by political rather than geographical barriers. This political fragmentation had another unexpected genetic consequence. It created unusual patterns of social mobility that influenced mate selection and reproduction. Unlike more centralized kingdoms, the German territories had permeable social boundaries, allowing individuals to move between social classes based on skill, education, and economic success. This social fluidity meant that genetic advantages could spread rapidly through the population, explained social geneticist Dr. Marcus Bauer. Traits that improved cognitive performance, technological innovation, or commercial success increased reproductive opportunities in ways not seen in more rigidly stratified societies. Evidence for this pattern comes from genealogical records showing unusually rapid expansion of certain family lineages across social boundaries. Families that began as peasants could, within a few generations, place members in scholarly, commercial, and even minor noble positions, increasing their genetic contribution to future generations. This genetic social mobility created distinctive selection pressures favoring cognitive traits, innovative thinking, and social adaptability. While these traits were valuable everywhere, the German political landscape provided unique opportunities for them to influence genetic representation in subsequent generations. As we move into the early modern period, German genetic distinctiveness takes another turn. The Reformation, which began in Germany with Martin Luther, created new social structures and marriage patterns that further influenced genetic selection. Protestant regions abolished monasteries and nunneries, returning thousands of educated individuals to the reproductive population. This sudden shift expanded the genetic pool in ways not seen in regions that remained Catholic. The dissolution of monastic life had profound genetic consequences, noted Dr. Hoffman. Individuals who might have removed themselves from the reproductive population instead contributed their genes to future generations, including genes that may have predisposed them to scholarly, contemplative, or independent thinking. Studies comparing Protestant and Catholic regions within Germany show subtle but significant genetic differences that emerged after the Reformation. Protestant areas developed higher frequencies of certain cognitive-related genetic variants, possibly reflecting this return of monastic individuals to the breeding population. The 30 Years' War, 1618 to 1648, created another genetic anomaly. This devastating conflict reduced the German population by up to 40% in some regions, creating a massive population bottleneck. Genetic studies of modern Germans show the signatures of this bottleneck: reduced genetic diversity in certain markers coupled with unusual frequency of rare variants that happen to survive. The war created a genetic founder effect, explained Dr. Fisher. Certain rare genetic variants that might have disappeared through normal population processes were instead amplified in the recovering population, creating distinctive genetic signatures that persist today. This historical bottleneck helps explain why certain rare genetic disorders appear at higher frequencies in German populations. It also created opportunities for rapid natural selection, as the post-war population expanded into emptied territories under the novel selective pressures of a devastated landscape. The ensuing centuries saw new dimensions of the German genetic mystery emerge. The Industrial Revolution, which transformed German territories earlier and more thoroughly than many other European regions, created novel selective pressures related to urban living, industrial toxins, and changing disease landscapes. Industrialization was a massive selective event, noted environmental geneticist Dr. Weber. Populations adapted to rural agricultural life suddenly faced coal smoke, chemical exposures, crowded living conditions, and entirely new patterns of work and diet. Genetic studies of industrial-era remains show accelerated selection for detoxification genes, immune responses to urban pathogens, and metabolic adaptations to processed foods. While industrialization affected all European populations eventually, the particularly intensive and early German industrialization created distinctive selective pressures. This industrial genetic selection might explain some of the unique disease resistance patterns observed in modern German populations, including unusual responses to certain pathogens and environmental toxins. It might also account for metabolic adaptations that influence how German populations process modern diets. The mystery deepens further when we consider the genetic impact of German emigration patterns. Between 1820 and 1910, more than 5 million Germans emigrated to the United States alone, with millions more settling in Eastern Europe, Russia, South America, and Australia. What makes this emigration genetically significant is its selectivity. Genetic studies of German diaspora populations reveal that emigrants carried distinctive genetic signatures, not representative of the general German population, but skewed toward particular regional and trait-associated markers. German emigration wasn't random, explained migration geneticist Dr. Stefan Klein. It selected for individuals with particular psychological traits, risk tolerance, openness to new experiences, and comfort with uncertainty. These traits have genetic components. This selective emigration created a feedback loop in the German homeland, altering the frequency of certain genetic variants related to personality, risk assessment, and novelty seeking. The remaining population showed increased frequencies of genes associated with traditionalism, caution, and local attachment. Yet paradoxically, Germany continued to show remarkable technological and scientific innovation despite this apparent selection for traditionalism. This contradiction suggests that the relationship between genetics and cultural outputs is far more complex than simple determinism. We might be seeing the results of cultural institutions that maximize the impact of innovative individuals even when they're statistically rare in the population, suggested Dr. Hoffman. German educational and research traditions may have amplified the contributions of genetic innovators even as their relative frequency decreased. This pattern of institutional amplification of rare genetic variants might explain another German genetic mystery: the unusually high frequency of certain neurodevelopmental variations associated with both cognitive challenges and exceptional abilities. Studies have found that certain neurodevelopmental genetic variants occur at higher frequencies in German populations than elsewhere in Europe. These variants, while sometimes associated with conditions like autism spectrum disorders, are also linked to exceptional abilities in mathematics, music, pattern recognition, and systematic thinking. There seems to have been some form of balanced selection, proposed neurogeneticist Dr. Julia Richter. These genetic variants bring both challenges and benefits, and something in German cultural or environmental contexts may have tipped the balance toward preservation rather than elimination of these variants. This balanced selection might help explain Germany's historical strengths in fields requiring systematic thinking and pattern recognition, from music and mathematics to engineering and chemistry. The cultural value placed on these cognitive styles may have created social niches where individuals carrying these genetic variants could thrive and reproduce. The modern era brings new dimensions to the German genetic mystery. Following the devastation and population displacement of World War II, Germany experienced one of Europe's most dramatic demographic transformations. Millions of ethnic Germans expelled from Eastern Europe resettled in a dramatically reduced German territory, creating new patterns of genetic mixing. The post-war population movements essentially reshuffled the German genetic deck, explained historical geneticist Dr. Wagner. Regional genetic signatures that had developed over centuries were suddenly dispersed across the country, creating novel combinations and accelerating the breakdown of historical population structure. This genetic reshuffling might have eliminated some of the distinctive regional adaptations that had characterized German genetics for millennia. Yet studies of modern Germans still reveal unusual patterns of genetic variation that distinguish them from neighboring European populations. Most notably, certain immune system gene complexes show distinctive patterns in German populations, possibly reflecting the unique pathogen histories and environmental exposures of Central European populations. These immune variations might influence how German populations respond to modern diseases, vaccinations, and environmental challenges. The contemporary chapter of German genetic mystery involves the integration of new immigrant populations. Since the 1960s, Germany has welcomed millions of immigrants from Turkey, the former Yugoslavia, Southern Europe, and more recently, the Middle East and Africa. Genetic studies of these immigrant communities and their descendants reveal something intriguing: accelerated genetic adaptation to the German environment. Within just a few generations, immigrant lineages show selection for genetic variants that help them process the local diet, resist regional pathogens, and thrive in the German climate. This rapid adaptation suggests that whatever environmental factors shaped historical German genetics continue to operate today, noted Dr. Brenner. We're watching in real time as these selective pressures influence newly arrived populations. This contemporary adaptation provides a unique opportunity to identify the specific environmental factors driving German genetic distinctiveness. By tracking which genes show evidence of selection in immigrant populations, researchers can pinpoint the exact selective pressures at work. Preliminary findings suggest that water chemistry, specific regional microbes, and dietary components unique to Central Europe may be among the key factors driving this adaptation. These environmental influences seem to target genes involved in metal metabolism, immune regulation, and neurotransmitter production. The question of why these environmental factors would be unique to Germany remains open. One hypothesis points to the distinctive geological history of the region, shaped by multiple glaciations, unique mineral deposits, and complex river systems that might have created a biological environment unlike other parts of Europe. Another hypothesis focuses on the historical intensity of human modification of the German landscape. Centuries of dense settlement, forest management, intensive agriculture, and early industrialization transformed the German environment more thoroughly than many surrounding regions, potentially creating novel selective pressures. Humans shape environments, which then shape human genetics, explained ecological geneticist Dr. Nina Schroeder. In Germany, this feedback loop may have operated with particular intensity for thousands of years, creating the distinctive genetic patterns we observe. This coevolutionary hypothesis gains support from studies showing correlations between historical land use intensity and genetic differentiation in European populations. Regions with longer histories of intensive agriculture and dense settlement, including much of Germany, show more pronounced genetic divergence from hunter-gatherer ancestral populations. This brings us to perhaps the most profound implication of the German genetic mystery. It challenges our understanding of human evolution as a slow, gradual process. The German case suggests that under certain environmental and cultural conditions, human genetics can change rapidly and dramatically, within centuries rather than millennia. We might need to rethink our timelines for human adaptation, suggested Dr. Dahl. The German evidence indicates that when the right combination of selective pressures exists, genetic change can occur much faster than we previously thought possible. This accelerated evolution might not be unique to Germany, but the particular combination of factors, geographical, historical, cultural, and ecological, has made it more visible and pronounced there than elsewhere in Europe. Germany has served as a natural laboratory for human adaptive potential, revealing capabilities that might exist in all human populations but rarely find the conditions to express themselves so dramatically. The final piece of our mystery involves the future implications of this distinctive genetic history. As Germany continues to integrate into an increasingly globalized world, will these unique genetic signatures persist, or will they gradually fade through admixture with other populations? Current evidence suggests a complex answer. Certain adaptive genetic variants, particularly those related to local environmental conditions, continue to show evidence of positive selection even in highly admixed modern populations. Other, more historically contingent variants are gradually declining in frequency as population barriers dissolve. We're likely seeing the difference between environmental adaptations and historical accidents, explained population geneticist Dr. Fischer. Variants that genuinely help people thrive in the German environment will persist regardless of ancestry, while those that merely reflect historical population bottlenecks will gradually disappear. This ongoing evolution offers a unique window into human adaptive processes in the modern world. By monitoring which German genetic signatures persist and which fade, we can better understand which aspects of our environment continue to shape human evolution even in our technologically modified world. The German genetic mystery ultimately reveals something profound about human evolution. Our genetics are shaped not just by our biology but by our history, our culture, our technology, and our social structures. The unique confluence of these factors in Germany created a genetic landscape unlike any other in Europe, a landscape that continues to evolve today. As we complete our journey through this genetic mystery, we return to the fundamental question: what makes German DNA so uniquely enigmatic? The answer, it seems, lies not in any single factor, but in the extraordinary intersection of geological uniqueness, historical contingency, cultural practices, and human innovation that characterize the German experience. From the mammoth-preserving Ice Age populations to the industrially adapted modern Germans, from isolated medieval principalities to globally connected contemporary communities, the German genetic story reveals the remarkable adaptive capacity of human beings when faced with novel challenges and opportunities. In the end, perhaps the most important insight from this genetic mystery is not about Germans specifically, but about human potential. The distinctive German genetic landscape demonstrates how quickly humans can adapt to new conditions, how profoundly our cultures shape our biology, and how the boundary between nature and nurture dissolves upon closer examination. As we face global challenges that will undoubtedly create new selective pressures, climate change, technological transformation, novel pathogens, the German genetic story offers both caution and hope. Caution that human populations can be dramatically transformed by their environments, and hope that our species possesses remarkable adaptive capacities waiting to be expressed. The true mystery of German DNA, then, is not just why it developed its distinctive patterns, but what those patterns reveal about the extraordinary evolutionary journey that all humans continue to undertake. A journey written not just in our cultures and technologies, but in the very molecules that make us who we are. In our genes, as in our histories, we find not just who we have been, but glimpses of who we might become, a story still being written with every generation in Germany and beyond.